Good morning, children. We are going to discuss about today antigens. In immune system, we are going to discuss about antigens. The term antigen, the term antigen. Okay, the term antigen, what is called antigen, is used in two senses. What is called two senses. Number one, the first to describe a molecule which generates an immune response and the second a molecule which reacts antibodies. In general, antigens are large, complex molecular substance large molecular complex substances that can induce a what you call detectable immune response. Okay, children, this is important. Thus an antigen is a substance that is specific to an antibody or a T cell receptor and is often used a synonym for immunogen. Okay, synonym for immunogen. Next one is an immunogen. Okay, an immunogen is a substance capable of initiating an immune response. Next point. Haptens. We call haptens. Haptens are substance that are non-immunogenic but can react with the product of a specific immune response. Next one, substances. Next, substance. Okay, immune response. Substances that can enhance the immune response to antigen. Okay, very very important. Are called adjuvants. What are called? Adjuvants. Next, epitope. Okay, epitope is an antigenic determinant and is the active part of an antigen. Next, a paratope. Okay, is the antigen binding okay site and okay and is a part of antibody. It's a part of antibody which recognize okay and binds to an antigen Okay children, this is important. Antigen. Next, the types of antigens. Types of what we call antigens. Types of antigens we are discussing. Now, on the basis of origin, okay, antigens are recalled. Antigens are classified into what we call antigens are classified into exogenous antigens and endogenous antigens. Exogenous antigens and endogenous antigens. Next, another important one. The antigens. Next one. The antigens which enter. Okay, which enter the host from the okay host from the outside, from outside in the form of microorganisms. Okay, microorganisms, pollens, drugs. Okay, pollens, drugs, or pollutants. Okay, pollutants. Okay, pollutants are called exogenous antigens. Exogenous antigens. Next, the antigens. Which are formed within the what you called within the individual or endogenous endogenous antigens. The best examples are okay, blood group antigens. Okay, children, observe very carefully what we are going to discuss. Okay, so first, the term. The term antigen is used in two senses. How many senses? Two senses. The first to describe a molecule which generates an immune response. So immune response, a molecule which will generate the immune response. And the second, a molecule which reacts the antibodies. Immune response, second one antibodies. In general, the antigens are large complex molecular substances that can induce a detectable immune response. So that is induces or that is detectable immune response. Thus, an antigen is a substance that is specific to an antibody. So one antigen have one specificity to one particular antibody. Or a T cells are receptor and often used a synonym immunogen. What you call? So very important. Thus an antigen is a substance that is specific to an antibody or a T cell receptor. Either antibody or T cells receptor, and is often used synonym for immunogen. Antigen, synonym immunogen. Both are same. Next, an immunogen is a substance capable of initiating. An immunogen is a substance capable of initiating immune response. The haptens. So immune response. Haptens means what? Are substance that are non-immunogenic but can react with the product of a specific immune response. Hapten are substance that are non-immunogenic, not at all immunity production, but can react with the product of specific immune response. Next, haptens are also called incomplete antigens. Next, the substances that can enhance the immune response to antigen called adjuvants. The substance that can enhance the immune response to antigen. Okay, the substance that can enhances the immune response to antigen are called, so what is called adjuvants, which enhances the immune responses to antigen. Okay, that is called adjuvants. Epitope is an antigenic determinant and is an active part of antigen. Epitope, where it is present on antigen, because of this only it will react with the antibodies. Antigenic determinant site and is active part of antigen. A paratope is the antigen binding site where the antigen go and bind to the antibody or not through this paratope. Antigen is binding to antibody. So is the antigen binding site and is part of antibody, which recognize and binds to antigen. Next, types of antigens. On the basis of origin, antigens are classified into number one, exogenous antigen, endogenous antigens. The antigens which enter the host from the outside, in the form of microorganisms, pollens, drugs, pollutants are called exogenous antigens. The antigens which are formed within the individual, which are formed within the individual are endogenous antigens. So in the body only the antigens are forming, in the sense they are called endogenous antigens. Best example, blood groups and antigens. Blood group antigens. Okay, blood group antigens. Okay, children, observe one more time. So antigen is used to in two senses. The first, to describe a molecule which generate an immune response. When enter into the body, immediately immune response will be takes place. That is called antigen, or a molecule which reacts antibodies are called antigens. In general, antigens are what? Large complex molecular substances that can induce the detectable immune response. Thus, an antigen is a substance that is specific to an antibody or a T cell receptor and is used as synonym for immunogen. An immunogen is a substance capable of initiating an immune response. An immunogen is capable of initiating immune response. Then haptens are incomplete antigen substances that are non-immunogenic but can react with the product of immune response. Substance that can enhance the immune response to antigen are called. So immune response will be develop and reactive towards to the antigen. Okay, enhances the immune response to antigen is called. Which one is a substance? Enhances the immune response to antigens is called adjuvants. Epitope is an antigenic determinant and is active part of the antigen. Paratope is an antigen binding site and is a part of antibody, which recognizes and binds to the antigen. Types of antigen. On the basis of origin, antigens are classified into exo from outside, endo inside. The antigens which are enter the host from the outside in the form of microorganisms, pollens, drugs are antigens, pollutants are antigens are called exogenous antigens. The antigens which are formed within the individual are called endogenous antigens. The best example is blood group antigens. The best example is blood group antigens. The blood group antigens. Okay, that point you should have to remember. Children, this is the most important point to remember. Next, we are going to discuss about antibodies. 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 Antibodies are immunoglobulin. Antibodies are immunoglobulin Ig. Okay. Protein molecule synthesized on exposure to antigen that can combine specifically with the antigen. Okay, whenever pathogens enter our body, okay, pathogens enter our body, the B lymphocytes produce an army of proteins called antibodies. Next, this is the most important, very important point. To fight with antigen. To fight with them means antigens. Next, very important point. Thus, they are secreted in. They are secreted in. Okay, thus they are secreted in response to antigen. Okay, response to an antigen by the effect of okay, B cells called, what is called? B cells called plasma cells. So plasma cells only produces antibody. The antibodies are next. The antibodies are classified into 5 major categories. Okay, based on their physiological and immunological, biochemical process or properties. Okay, this is the important point. Next, they are IgG, gamma. Next, okay, gamma, IgM, mu, IgA alpha, IgT delta, IgE epsilon, called epsilon. Okay, children, this point you should have to remember. IgM, IgG, IgA will be there. Okay, children, observe very carefully. Okay, antibodies are immunoglobulins, protein molecule synthesized on exposure to antigen. Suppose antigen entered, antibody will be formed that can combine specifically with the antigen. Specificity. Whenever pathogen enter our body, the B lymphocytes produce B lymphocytes. This is important. Whenever pathogen enter in our body, what will happen? The B lymphocytes produce an army. Army means nothing but fight for enemies, fight with enemies. Okay, called army of proteins called antibodies to fight with them. Thus, they are secreted in response to antigen by the effect of B cells called, which one? What kind of B cells? Plasma cells. What do you call the cells? Plasma cells. Antibodies are immunoglobulin, Ig, protein molecule synthesized on exposure to antigen that can combine specifically with the antigen. Whenever pathogen enter our body, the B lymphocytes produce an army of proteins called antibodies to fight with them. Thus, they are secreted in response to an antigen by the effect of B cells called plasma cells. The antibodies are classified into five major categories based on their physiological biochemical properties. What are the major categories? IgG, immunoglobulin gamma, IgM, immunoglobulin mu, IgA, immunoglobulin alpha, IgD, immunoglobulin delta, IgE, immunoglobulin epsilon, IgG, immunoglobulin gamma, IgM, immunoglobulin mu, IgA, immunoglobulin alpha, IgD, immunoglobulin delta, IgE, immunoglobulin epsilon. Like that, there will be types of antibodies are there. In 1950s, okay, 1950s, experiments, okay, 1950s, experiments by Porter and who is the scientist? Edelman, Porter and Edelman. Okay, children, Porter and Edelman revealed the basic structure of the immunoglobulin. Okay, children, an antibody molecule. Next, an antibody molecule. Okay, an antibody molecule. Okay, which shaped an antibody molecule is Y-shaped structure. Regard Y-shaped structure. It is Y-shaped structure. Okay, children, it's a Y-shaped structure that comprises okay, this point you have to that comprises okay of four polypeptide chains. Okay, two identical light chains, okay, two identical light chains, L of molecular weight, okay, children, the molecular weight to 25,000 Daltons. Okay, next, approximately okay, 214 amino acids. Approximately 214 amino acids and 2 identical heavy chains, and 2 identical heavy chains are there. So 2 identical heavy chains. Okay, H. The molecular weight is 50,000 Daltons. Molecular weight is 50,000 Daltons. Approximately 450. Record 450 amino acids. Okay, children, observe very carefully. We are going to finish this. In 1950s experiments, Porter is a scientist. Porter and Edelman revealed the basic structure of the immunoglobulin or antibody. An antibody molecule is which shaped? Y-shaped molecule structure that comprises 4 polypeptide. 4 polypeptide means amino acids, protein chains, 2 identical light chains, light chains. Molecular weight is 25,000 Daltons, approximately 214 amino acids. And 2 identical heavy chains, its molecular weight is 50,000 Daltons, approximately 450. Light chain, 25,000 Daltons, 214 amino acids. Heavy chains, 50,000 Daltons, molecular weight, 450 amino acids. Still, some more points are there. In the tomorrow's class, we will discuss about a complete structure about the antibody. Thank you, children.